Welcome back to Biology 102, and today I want to wrap up meiosis with talking about 1, how mitosis and meiosis are different, 2, how meiosis gives us some, variation within our population, and 3, some problems with meiosis. So here I am displaying a table and it shows sort of the comparison between mitosis and meiosis. Notice that S phase in DNA replication occurs the same before each of these cells starts division in interphase, so that is very similar. Both of these processes will start with a diploid cell, but then things start to change, right? So mitosis, you're going to start with one diploid cell. You're only going to do one round of division versus meiosis that does 2. And you're going to create 2 different kinds and numbers of cells. So in meiosis, you're going to create 2 identical diploid cells. In meiosis, you are going to create 4 haploid cells that are unique from that parent cell. And the role in the body is quite different. So in meiosis, we do mitosis to help us grow, to help us replace and repair old cells. So there's lots of good reasons why we do this every day. Meiosis is really about producing gametes. So producing sex cells, getting some variation within our population so that we have sort of offspring that are different than our parents. Now, if we look at this summary, make sure that you know some similarities and differences between mitosis and meiosis. The second important point about meiosis is the fact that it gives us genetic variation. And we get genetic variation in 3 different places in meiosis. We get it crossing over in prophase 1. We get genetic variation when our chromosomes line up randomly on the metaphase plate in those tetrad sets at metaphase 1. And we also get variation from just random fertilization, like which sperm joins with which egg so that we get that first cell. Now, I want to sort of walk you through each of these just so that you have an idea of how this works. So crossing over, this is where we crossed over equal sections of these homologous chromosomes so that equal pieces of DNA are exchanged between the paternal and maternal chromosomes. Enzymes then will proofread this, but you can imagine that this gives us some variation. If you have a grandmother with brown hair and brown eyes and a grandfather with blonde hair, blue eyes, then this sort of mixing up of the genome can give you offspring with brown hair and blue eyes, combinations of traits that we haven't seen before. So this is one place that we get genetic variation. We also get genetic variation from random assortment and arrangement of chromosomes on the metaphase plate. Look over here at these possibilities. You could have it where all the paternal blue, mostly on arm one side, this will sort into mostly the blue chromosomes, right, in your sperm and eggs, and then or the red in sperm and eggs, about half and half. Or you could have possibility 2 where you have them mixed up and then you get combinations of blue and red in each of your sperm and eggs. So how these chromosomes actually line up at metaphase in the middle of the cell makes a difference and gives us some unique gamete possibilities. Lastly, the race of the sperm. So just after meiosis, in order to create a fertilization event and create this first zygote cell, then the sperm actually race to the egg and join with it. If eggs have 2 different positions or 2 different chromosomes that can line up in 2 different ways along the metaphase plate, then eggs in a human create, without considering crossing over, 8 million possibilities, and sperm also 8 million possibilities. So for each couple, there is 64 million possibilities of sperm and eggs and their combinations, again, within sort of one couple. So this gives us a lot of possibilities, and this is without bringing in crossing over. That gives us even more. This is why siblings don't look the same. They don't act the same, right? They don't have the same traits, behaviors, etc., because there's so many possibilities between these sperm and eggs. The last point I want to make about meiosis is a common problem. It's called non-disjunction, where members of your chromosome sets or your tetrads, they separate or fail to separate in your final sperm and eggs. This leads to things like Down syndrome. Down syndrome, you have an extra 21st chromosome, or Klinefelter syndrome, where you have long, short. So you have an extra chromosome in that 23rd position. Turner syndrome is where you're missing that second chromosome. So you have long and then a missing chromosome. So thanks again for watching our comparison of mitosis and meiosis and common problems with meiosis, and I'll see you again next time.